All right, and so the next little bit is really more of a bit of a background that will get you started with your final assignment. So the assignment is going to go into a lot more of the detail. So that's where kind of most of the information will come from is completing your the final assignment. But just to give a little bit of information about some of that content. So particularly this concept of a membrane potential, and then how our neurons actually send these messages. So how these electrical signals are generated and propagated down the neuron. So in this figure here, so this is taken from an external source here. This is a nice way to kind of summarize in a single slide. You're more than welcome to use your textbook to kind of help supplement some of this material, and maybe you'll find as you're going through the assignment that might be of benefit to you, but we're going to use this one for now. Right, so on the right side of the figure here, which is our intracellular, this is going to be, say, like inside the neuron. So let's just say this is an axon, right? So this is going to be inside the axon on the right side here, and then on the left side is going to be the extracellular space, right? So inside on the right, outside on the left. And what we see is kind of both inside and compared to outside are going to have different chemicals with different charges. So these are going to be, remember, charged particles are called ions, right? So there's going to be a different proportion of ions inside and outside the cell. And so as we can see, the inside is going to be more negative than the outside. So we have, right, our negative charge inside, and we'll have a positive charge outside, right, comparing the two to each other there. So all this is showing is that things like kind of these larger proteins that have a negative charge and chlorine, right, are going to outnumber the positive charges that we see. Then compared to the extracellular on the outside, right, we're going to see much more of these positive charges in the extracellular space. These arrows, right, so the arrows that we see here, the three of them, these are our channels, right? So these are our channels. We're going to regulate the flow of these ions across the membrane. So when we're resting, all of these channels are closed, and we have this this intracellular, extracellular, negative on the inside, positive on the outside. It's kind of our neutral starting point, if you will. Not neutral because of the charge, but that's our starting point. So what happens is when a neuron is going to get a signal to tell it to send a message, right? So a neuron is getting a signal that says, okay, we need to send a message, send an impulse, an electrical impulse, which is how how our neurons send those signals, right? So we need to send this electrical signal down our axon to the to another either another neuron or to an effector. So what happens is the channels are going to open up, and the charged particles are going to kind of move based on where they want to go, right? So you'll see in your assignment there's going to be talking about a chemical gradient as well as an electrical gradient. And so often what we'll see is when we're about to send our signal, so these channels open up, and what we end up with is a lot of, in particular, sodium, which we see on the outside, is going to want to flood into the cell. Right, so we get a bunch of sodium that's going to be passing through these channels that are now open. So they're going to move from outside to inside. And what this does is this is going to make the inside of the cell less negative, right? So as our positive sodium is flooding into the cell, now the inside of the cell is becoming less negative or more positive, right? Because sodium has a positive charge, and what this does is then going to kind of create that that change in the electrical signage, basically. So in this region here, so this entire region of the cell, right, we would then have this this inside having a different charge, and then what happens as these channels open and the ions move in, right, that's kind of what's going to create that electrical signal as this inside becomes less negative. So this change in location is then going to flow down the membrane, and it's going to basically open up other channels along the way, which is then going to influx sodium into the cell, right? Again, so we have that change in membrane potential, and this is going to continue that process of moving down the membrane. Eventually, as these move away, right? So as the as this electrical signal is moving away, these channels will then close, and then we will kind of recalibrate and go back to our normal resting potential with the more negative on the inside. So what this leaves us is basically there's two types of kind of membrane potentials. So we have there's the resting membrane potential, right? So we have resting, which is what we are basically starting with, right? So resting membrane potential, all the channels are closed, and we have that more negative on the inside. But when we have the channels open, we have what's called an action potential, right? So an action potential, I still don't know how to write at this mouse apparently. Action potential, that's a no. And so it's the action potential, which is that kind of that flow of the charge, that difference in charge down the membrane. Right, so the channel's open and ions flow across the membrane, which make the inside less negative, which generates that action potential. So again, you'll see a lot of this in much more depth and probably as a better explanation as you go through your assignment. I just wanted to introduce kind of some of the basic concepts here, though. So we end up with what's called a synapse, which is basically kind of that connection point. So it's where the nerve cells are going to talk to each other or talk to some sort of effector, right? So when one neuron passes its nerve impulse to another neuron or effector cell, right? So that point of, I don't want to say contact, because it's not really touching, and we'll see that coming up, but it's that it's kind of where the signal is going to pass from one to another. So we see three types of neuron to neuron synapses in particular. So when one neuron is speaking to another one, we can see three types of synapses. So the first is called axodendritic, right? So an axodendritic synapse is going to be the axon of one nerve cell, axon of one neuron, synapsing with the dendrites of another, right? So that's the axodendritic. We have the axosomatic, which is basically going directly to the cell body, right? So the axon of one neuron isn't going to synapse with the dendrite. It's going to go just directly to the cell body itself. And then finally we have the axonal, right, which is when the axon of one is going to synapse directly with the axon of another, right? So we're going to essentially just bypass the cell body at that point and just go right to the other axon, although it is kind of done near the cell body, right? So it's not just going to be in kind of the middle. We'll have the myelin sheaths that are going to be in the way. So it's kind of right where the axon is starting to branch out from the cell body is where we'll have this axonal type of synapse. Okay, and there's two types of synapses. The most common one is called a chemical synapse, and so that's what we're going to just take a look at very briefly here. So what happens is we end up with what's called the presynaptic neuron. And so this is the neuron where the electrical signal is coming from, right? So we can see here the action potential, right, is flowing this way down the presynaptic neuron. And then it's going to, basically electrical signal is going to create a chemical reaction. So it's going to, don't worry too much right now about the different kind of voltage gated calcium and what that means. But essentially you get this influx of calcium, which is then going to release what's called a neurotransmitter into the synaptic cleft. So the synaptic cleft is the small gap between a presynaptic neuron and a postsynaptic neuron, which is basically the neuron that is receiving the signal. Right. So mentioned in the last slide, they're not actually in direct contact. They're not touching. Right. So there's going to be this very, very tiny gap, right, which is called the synaptic cleft. And it's within the synaptic cleft that we have these neurotransmitters, right, which is basically a chemical signal that is going to then interact with the postsynaptic neuron and continue the action potential flow. Right, so we have the nerve impulse. So nerve impulse is the same as an action potential. You might see them used interchangeably in some of the different study tools. So we have the nerve impulse coming from the presynaptic neuron. It's going to basically cause the release of what are called neurotransmitters. These neurotransmitters are going to cross the synaptic cleft and then enter into the postsynaptic neuron, which will then create the action potential at the postsynaptic neuron to continue down its way. All right, so I mentioned before there are two types of synapses. There's the chemical and an electrical. So chemical is really the most common type. You really only see electrical synapses in in a developing fetus while they are still developing, and then eventually they will convert to the chemical type. So that's why we really see the chemical as this most common type of synapse. And so that is that release of the chemical messengers called those neurotransmitters. So what we were looking at in the previous slide is a type of chemical synapse. The electrical synapse mentioned before, right? So very rare, mostly just in the developing fetus, and that's where we have this kind of direct between neuron transmission. So it's that kind of direct transmission. So one way to kind of think about these two different types. So a chemical synapse is kind of like two people shouting a message across a lake, right? So it's effective if you're on one side of not like one of the Great Lakes, but if you're on one side of a lake and shouting across to someone on the other side, it's effective. It's going to cover a long distance, right? Sound's going to carry over water, but it's going to be slower, right? So it's not going to be as fast. And this is the type of synapse that occurs, either between neurons, so neuron to neuron, or between the neuron and an effector, so something like skeletal muscle. Right? So that's where we have those types of synapses, like shouting across the lake. On the flip side, right? So the electrical is basically like acting like a doorway, right? So as instead of shouting across a lake, you're basically just walking through a door within the same house to pass the message across. So it is much faster, right, in terms of that speed of transmission. So you can picture it's it would be slower to get the message to someone when you have to shout across a lake versus if you could just open a door and talk to the person directly there. And so that's why the electrical is much faster, but again, more rare and only that we see in this kind of developing fetus until it converts to the chemical type of synapse. So most synapses, if not all, once we get to kind of age, right, is going to be this chemical type here. Okay, and so that is where we're going to end today. You should have enough information now to go ahead and start the assignment, which will hopefully clarify some of the latter half of this lecture, particularly talking about the memory potential in the different synapses. And then next week, we'll wrap up by taking a look at these two different different types of the nervous system, so the central and the peripheral. So yeah, thanks for listening. As always, if you have any questions, feel free to post them in the discussion forum, and we'll see you soon. Right, bye.